Streaming on YouTube, very impressive, as well as to Allison off in New York. So I'm Matt Harms, and it's my distinct honor to co-chair Janet's thesis dissertation presentation today. The other co-chair is Allison Goat, who's live via video conference at Mount Sinai, where her lab is located now. Because Janet is the first of hopefully many graduate students in my lab, I'm especially indebted to Dr. Goat in particular for co-mentoring Janet with me. And a lot of the credit for the brilliant insights that I had and the wonderful leadership that I was able to provide came directly from her. Either that or from Tim Miller or the other members of her committee who joined us today, and I'm very grateful for that. So I also wanted to acknowledge Terry and David, Janet's parents, who have come to join us today to share in Janet's presentation. And then I'd like to invite each of you to also join us for a reception at 4 o'clock in the first floor conference room over in biotechnology to help celebrate the work that Janet has done. So I think that each of you over the course of her presentation today will be impressed with the amount of work that she's managed to accomplish during her her doctoral work. That's right. So it's probably even more impressive when you realize that there have been quite a few obstacles, external obstacles that Janet has had to overcome. Not the simple, the method's not working, the science isn't going well type of obstacles, but obstacles in the principal investigator lab leader department. So just as Janet was getting ready to propose her thesis, her first mentor, Bob Balo, moved to Cedars-Sinai in Los Angeles. And Dr. Goat and I assumed responsibility for mentoring her. Almost as soon as that occurred, my family experienced a tragedy. And so for the next 2 years, I spent more time over at Children's Hospital than actually in the office, giving Janet the guidance and things like that that one might have hoped. But despite that, she persevered, had lots of creative insights and directions to go on her own, and has generated quite an impressive body of work that I'd like for her to present to you now. So without further ado, here's Janet King.
All right, thank you, Matt. Today I'm going to be talking to you about the genetic factors that contribute to the pathogenesis of amyotrophic lateral sclerosis. A lot of you probably aren't really sure what that is. You might know it as the Gehrig's disease, or that thing from the Ice Bucket Challenge. So what it is it's a fatal neurodegenerative disorder that affects the motor neurons in the spinal cord and in the motor cortex that leads to muscle weakness and wasting, paralysis, and eventually death due to respiratory failure. And onset can either be in the arms and legs, and we'll refer to that as spinal onset, or it can start in the bulbar onset with difficulty with speech and swallowing. So here's just kind of the makeup of our kind of a conglomerate cohort of different studies that we used in our lab. So you can see it's a little bit more common in men than it is in women. Our cohort here in North America is predominantly white, non-Hispanic, of European ancestry. And age of onset median is about 60, but as you can see, there's a really wide range, and we do have some people that have juvenile onset, as early as age 14. It's most commonly onset is in the spinal area, so affecting the arms and legs first, and then bulbar is the most common. And some people, have less common forms that start with frontotemporal dementia or just a diffuse degeneration. Survival is about 30 months after age of onset. And again, you can see there's a really wide range in that. And we're not going to really get into the factors that contribute to that very much today. So the families that have ALS have been very valuable in providing genes that can cause disease. 40% are caused by repeat expansions in the C9orf72 gene, which you can see over here. This GGGGCC repeat in between two alternate untranslated 5' UTRs. So in the transcript variants, it can either be upstream or in the non-coding intron. And that contributes to about 40% of familial ALS cases. The next most common are point mutations in SOD1, accounting for about 20%, and then TDP, target DNA binding protein, and I'm going to use them interchangeably with TDP43, so those are the same thing, and FUS are each about 5%. And then there are about 20 additional genes, but each of them accounts for only a small portion of cases. These families have been vital in providing insights into how ALS happens, how motor neuron degeneration happens. And here's just kind of a list of some of the pathways that are involved. You've got apoptosis, mitochondrial dysfunction, impaired axonal transport, ER stress, misfolded proteins, altered RNA processing. So finding additional genes is hopefully going to help us narrow down which of those pathways are really, causing disease and which ones are just side effects of other impaired processes. So that's what we learned from families with ALS, but most people with ALS don't have a family history of disease. Oh, my clicker's not working. Okay, so about 90% of cases don't have a family history, and we consider these sporadic or SALS. And some of these can be attributed to some common issues like non-paternity, where the parents aren't actually the parents. And so maybe the real parents had ALS. Some of it can be due to small family size. ALS is onset in later life, so if you have a smaller family, it's more likely that more of them won't live to the age where they would have developed symptoms. And there's also the issue of incomplete penetrance, where not everybody with a certain mutation will actually get disease. So a few studies have looked at the genes that cause familial cases in sporadic ALS. And again, we see C9orf72 expansions are the most common cause, again, but only contributing about 6%. And only really the most common familial genes have been looked at in sporadic cases. And you can see in this subset of studies that there's a lot of heterogeneity in the amount that they contribute. SOD1 is typically the most common, about 2%, 1 to 2%, but then we have this study in Ireland where it's 0. So that makes it really difficult to confirm findings in additional cohorts when you have that amount of heterogeneity across populations. Recently, this idea of oligogenic inheritance has been studied, and I'll talk a lot about this later, but the basic idea is that you have to have mutations in more than one ALS gene to develop disease. So, for instance, in this pedigree, they have a mutation in angiogenin and TDP. And so a couple of studies have reported this, so this is something that we look at as well. There are a few risk factors in sporadic disease. One of which is intermediate-sized expansions in the ataxin-2 gene. Normally, full expansions over 34 repeats cause spinocerebellar ataxia type 2. However, it's found that intermediate length expansions of about 20 to 30 repeats also increase the risk for ALS. There have been a couple of genome-wide association studies looking for risk factors, and they found a couple genes, but none of them have consistently been replicated. Again, that's because of the population heterogeneity. Maybe different populations have different risk factors. One type of risk factor that hasn't been looked at very much is rare variants. These are variants that are present in less than 1% of the population. And while individually rare, cumulatively they could explain a large portion of disease. So you can see on the right how we can have a common variant where it's pretty easy to tell this is much more common in ALS, the red lines over here, than in controls. But if it's very rare, we don't know if this difference we see is actually significant. But you can look across the entire gene, and maybe cumulatively, these mutations are more common than these mutations. So the goals of this study, I'm kind of going to give you a broad overview of 3 different ways we look at the genetic contribution of ALS. The first is, what is the contribution of these known genes that I talked about in our cohort? And then what characteristics of the C9orf72 expansion are important, since this is the most common cause of disease. And then can we identify novel genes that are causing ALS? So the first part, we performed targeted resequencing of our known ALS genes so we could understand their contribution to disease in our cohort and to investigate the oligogenic inheritance and the rare variants. So this is the approach we took. It's called pooled sample sequencing, where we combine pools of genomic DNA from patients together, and then from those pools amplify our genes of interest, and then repool them together and sequence them, and then use a program called Splinter that was developed here at WashU to align our reads and call variants. And then we have to go back and genotype those variants in the individuals to validate them and find out who they came from. So this is the list of genes we sequenced. And in addition to the sequencing, we looked for repeat expansions in C9orf72 and in Ataxin-2. So in total, we sequenced 349 sporadic ALS subjects and 42 familial ALS subjects, and identified 65 rare and novel coding and splice site variants. And these we broke down into 4 different categories based on how likely we thought there were to be actually pathogenic mutations. The first is category 1, which are reported in ALS. They're not present in any control databases, and we're pretty, there's functional evidence that they are actually causing ALS. So this included 6 mutations in SOD1 and TDP that we knew about going in. So those were our positive controls, and we were able to detect them all. And in addition, there were 2 FUS mutations and an additional TDP and a BCP mutation. Category 2, mutations that are reported in ALS, but have since been found in normal control databases. And we're not really sure, if those were erroneously reported as disease mutations in the first place because they didn't include enough controls to begin with, or if they're incompletely penetrant, or if the controls are actually not old enough yet to have ALS, and so they might go on to develop ALS. But a couple interesting mutations in here, one known recessive SOD1 mutation, and we did find one person that was homozygous for that mutation. And then also a possible recessive mutation in Senataxin, which had not had recessive inheritance reported in ALS before. Category 3 are mutations that are novel in ALS and are not present in databases. So this has included a splice site mutation in DAO and a de novo splice site mutation in a juvenile onset subjects in the FUS gene. And then nonsense mutations in FIG4 and octoneurin. Octoneurin has had several nonsense mutations reported before, and this falls in between those, and so it's also likely to be causing disease. And then a novel SOD1 mutation that's in the same amino acid as a previously reported SOD1 mutation. And then category 4 are ones that have not been reported in ALS, but are also rare in databases. So we don't really know yet if those are pathogenic or if they just are, along for the ride. So here's kind of the breakdown by gene. You can see that C9orf72 is the most, this is all the same mutation, the C9orf72 repeat expansion. It's the most common cause in both familial and sporadic. So we've found mutations in 64% of people with familial ALS and about 28% of sporadic ALS, which is higher than other previous reports. But that's likely due to the number of genes and the differences in pathogenicity criteria. We kind of took a broad, everything under 1% we're considering to be potentially pathogenic. And so if we look further by breaking down familial cases, by strength of familial inheritance, we have definite cases that have at least two first or second degree relatives with ALS, and about 80% of those have mutations in known ALS genes. And then as we get less and less, strength of familial inheritance down to, one distant relative had ALS and then sporadic, the contribution of these known ALS genes decreases because these have been, are familial ALS genes and therefore likely to be more penetrant and cause familial disease. So next we looked for subjects with variants in multiple genes. Here at the top, we actually have a couple recessive and compound heterozygous. So those are mutations in the same gene, but then we also have a lot of cases with mutations in more than one gene, including several people that had mutations in 3 genes. And we didn't sequence controls at the same time, so we weren't able to compare if this was statistically more common in ALS compared to controls. But based on other studies, it does look like it is more common. But even more interesting is that we found that people with mutations in multiple genes had earlier age of onset than people that had mutations in one gene, or even people that had mutations in no genes. And that the age of onset correlated with the number of genes affected, which was this really interesting, actual functional link between this oligogenic model and clinical characteristics. So next we looked for rare variants in our sporadic ALS population. We didn't find any single variants that were associated with ALS. So we used SCAT as a gene-based method of looking at the combined effects of rare coding variants across genes. And the only gene that passed our significance threshold was SOD1. But TDP and VAPB were close. So I think we're just going to need larger sample sizes to be able to really tell, to have the power to find significant associations for rare variants. All right, so summarize this section. We identified 65 potentially pathogenic variants in addition to the repeat expansions we looked at, and a total of 124 subjects, accounting for 31.7% of our total ALS cohort. But many of these variants are of unknown significance. We did see an earlier age of onset in subjects with mutations in multiple genes, which suggests that there's a synergistic effect between them. And then we saw a significant association with SOD1, but not with any individual rare variants. So next, we looked at characteristics of the C9orf72 expansions. So as I said before, these are hexanucleotide repeat expansion of the GGGGCC repeat unit. They're the most common cause of ALS. Individuals normally carry around 2 repeat units, but they can be up to about 30 repeats in normal subjects, while full expansions range in the hundreds to thousands of repeats. So some of the key questions we have about this is, why is this expansion so common in sporadic ALS? Why isn't it causing disease in everybody that has it? And how big do the expansions have to be pathogenic? The ones that we've seen are all very, very large, but is there some sort of threshold to when they actually become toxic? And similarly to that is, why aren't all tissues affected? This gene is present in every cell of your body, but for some reason only affects neurons, apparently. So to look at the first question of why is it so common in sporadic ALS, we asked the question, are all expansions derived from one event, or are there multiple events that can lead to the expansion? And in a single expansion model, that would imply that there are other factors, whether it be genetic, epigenetic, or environmental, that are contributing to the reduced penetrance of this mutation, and why this sporadic ALS, people that carry the mutation, why their parents didn't also have ALS. And then in a multiple expansions model, that would mean that they could possibly be arising spontaneously from pre-mutations as happens in other repeat expansion disorders. So all expansion carriers are known to share a common genetic background or haplotype, which originated in Finland. And this haplotype is marked by the SNP RS3849942. So this is the ERIS background for expansions. And this has been taken as evidence that there was a single expansion event. You can see in this model, this blue tick mark is representing the risk background. And the expansion arose on the background and then was passed down. And so all expansion carriers are descended from that one expansion event. But the other explanation is that this is just a background that is prone to expansion. So we have these, risk backgrounds in the population, and then they can develop expansions because of probably differences in DNA replication that make them more prone to instability. So first we looked at the known risk background in our cohort, and here are 8 different SNPs from that background that we genotyped, and this here is the tag SNP. And we can see that not everybody carries the entire haplotype, but the ones that don't carry the full haplotype can be explained by recombination events, which the recombination frequency is marked by this green line. And here the purple line is where the expansion lies. So most people carry at least part of this haplotype, and there was one subject that didn't carry the tag SNP, but they carried the SNP that was downstream of the repeat expansion, so that can also be explained by recombination. So to go a little more in depth, we decided to sequence a back that contains the repeat region from one of our patients. And we sequenced them by MySeq and identified 4 rare variants within this haplotype block, all of which segregated with the expansion of family, as they should, since they were derived from the chromosome that carries the expansion. So we decided to focus on this one, RS147599399, because it's about 600 base pairs away from the tag SNP, and so we thought this one would be the most informative to determine whether or not this is all, the repeat expansion has one single origin. So we genotyped this mutation in 106 individuals from 86 families that carried the expansion, and found 19 carried this SNP from an allele frequency of 11% versus 0.339% in 1,000 genomes and 1% in 1,000 genomes of European ancestry, which gives us a p-value of 1.625 times 10 to the negative 10. So it's very significant, very associated with the repeat expansion. And the variant segregates with the expansion in the 2 families that had multiple carriers that we tested. And as you can see, here's a breakdown of all of the different populations in the 1,000 genomes, and it is not present at all in the Finnish population where the risk haplotype is thought to have originated. It's actually most common in Italy. So here we go back to our models of expansion, where we can add in our new SNP and the green tick mark. So if it was a single expansion event, we would expect to see this risk background with the blue tick developing the expansion, and then the SNP developing after the expansion, and not creating a background, except in that instance, we don't see our new SNP in non-expansion carriers. And alternatively, we could have the SNP develop in non-expansion carriers, and then the expansion developing separately, but in that case, we don't see our new SNP in expansion carriers. So since we see the SNP in both populations, it is not likely to be a single expansion event, which leads us to multiple expansions, where we can have the expansion developing on a background, both with and without RS147599399. Or it can be kind of alternate version of that, where we have a pre-mutation in the general population, and those appear to be non-expansion carriers. That pre-mutation is more likely to develop into a full expansion. But either scenario leads to multiple events of full expansions. Interestingly, we looked at some clinical characteristics, and we found that expansion carriers that carry this rare SNP actually have prolonged survival compared to expansion carriers that don't. So you can see in this Kaplan-Meier curve, the median survival after onset in people with the SNP is about 52 months compared to 30 months in people that don't have that SNP. And we don't see this effect in non-expansion carriers. So it's an actual effect that this SNP is having to mediate the effects of the repeat expansion. So the next characteristic of the repeat expansion we looked at is their size and their stability. So how big do they have to be pathogenic? Why aren't all of the tissues affected? And what happens as they get passed down in families? Do they stay the same size, or do they continue to grow or shrink? So the way we detect repeat expansions is by repeat PCR, but those can't determine size of full expansions. They can only size expansions up to about 15 repeat units. So we have to use Southern blots to size full expansions. And so we have an overview of Southern blots, because no one does them anymore. First, we start with a restriction digest of our genomic DNA, which we run out on a gel to separate by size. And then transfer to a membrane, which we can add radioactive probe to. Then wash off the probe and expose it to the membrane to film, and the probe only sticks on our sequence of interest, and so we see bands where our repeat is. And so this is kind of a cartoon version of what we see. Is here we have our wild type. Everybody has one normal sized expansion, and then here we have our repeat expansions. So here's just an example of a blot from blood-derived DNA. We see these long smears, which we think are indicative of a range of repeat sizes. So that means that we've got some somatic instability of the repeat. We also looked at blood-derived DNA across families, and we kind of see a variety of different patterns. In this one family, here we have the proband, and proband's son and 3 siblings who are all unaffected, but they all have roughly the same size expansion. And this family is interesting because they actually there's a distant cousin that has ALS but doesn't have the repeat expansion, and you can see on the blot that is confirmed. But then that person's cousin also has ALS with a repeat expansion, and that's about the same size as the rest of the family. And then here we have a really interesting family where the proband and her unaffected daughter, mother, and brother all have larger expansions than her. And then we have other families, for instance, here is a proband with ALS and their identical twin who doesn't have ALS but has the exact same size expansion. So this is kind of indicating that either expansions in blood-derived DNA don't really mean anything, and that we need to be looking at more relevant tissues, brain tissues and spinal cord, or that there are environmental factors that are also contributing to the pathogenesis of ALS. So here's just a box plot showing our repeat sizes in blood-derived DNA in unaffected carriers, or affected carriers and unaffected carriers, and there is no difference between them. So next we looked at a variety of tissues. We noticed that in expansions in the brain were larger than expansions in the blood. So we took that further to look at different brain regions. Here's just a representative plot of some different tissues we looked at: the motor cortex, basal ganglia, cerebellum, brainstem, etc., and then also some non-neuronal tissues, including heart, liver, skin, and testes. One interesting thing we see is that this smaller expansion in the cerebellum compared to other neuronal tissues is consistent across all expansion carriers we've seen and that other people have also reported. So here's some, we weren't able to get very many tissues or very many people to get good blots from. So this is just the number of the people that we have blots for. So you can see cerebellum is the only tissue that really looks different from anything else, including kidney, which is unaffected in ALS, but it does not have smaller repeat expansions. So it's really telling us that the repeat expansion in the size is not the most important factor that determines whether or not the tissue will be affected. And then maybe other tissues or some tissues are better at dealing with the effects of the repeat expansion than others. But we'll need larger sample size to be able to confirm this. We also didn't see any correlation with expansion size in any tissue with any clinical characteristics. So to summarize what we know about C9orf72, we found a rare SNP in the risk background that shows the multiple origins of the expansion, which brings up the possibility of that de novo expansions can account for sporadic ALS, and that both the risk haplotype SNP RS3849942 and our new SNP RS147599399 are cis-acting modifiers of the expansion, with the former leading to a background that promotes expansion, and then the latter somehow mediating the effects of expansion. And this is supported, the idea of multiple expansions is supported by the instability that we see of repeats, both in families and in different tissues. And that you don't see any correlations between repeat size and clinical characteristics. So we need to look for additional factors that are contributing to pathogenesis. So the last part I worked on was identification of novel genes that cause ALS. So based on our full sample sequencing, roughly 35.7% of our familial cases and 72% of our sporadic ALS cases still don't have a genetic cause. So discovering additional genes that may contribute to those cases will also help us identify novel pathways that are involved in disease. So the one pathway that we do know that is involved in disease, however, is the microglia and neuroinflammatory pathways. So we decided to look at genes that are regulating microglial activation as potential candidates. Mainly the gene TREM2, which is known to cause an autosomal recessive form of early onset dementia called polycystic lipomembranous osteodysplasia with sclerosing leukoencephalopathy. And a rare variant, R47H in TREM2, has also been identified as a risk factor for Alzheimer's disease. Now, TREM2 forms a signaling receptor complex with another gene, DAP12, which is expressed on macrophages, dendritic cells, osteoclasts, and microglia, is involved in regulating microglial activation. So we genotyped the same R47H SNP in our ALS cohort and in controls. And since the SNP is different frequencies at different ages, we used a logistic regression, age and gender as covariates, and found that it was significantly associated with ALS, with an odds ratio of 7.38 and p-value of 0.0032. So we looked at expression of TREM2 in both S31 mice and saw a drastic upregulation in the G93A mice in spinal cords, and also an increase in TREM2 expression in spinal cords of ALS patients, which was even more drastic when we looked at just familial cases. So R47H was significantly more common in sporadic ALS, and has also been shown to be an Alzheimer's risk factor. So this points to neuroinflammation as kind of a common pathway between neurodegenerative diseases. This also provides a genetic link to the observed microglial involvement we see in ALS. I'm not going to show you this data, but we looked at mutations looked for other mutations in TREM2 and in other TREM genes. We didn't find any additional TREM2 variants associated with disease, and we found a possible association with the related gene TREM4. That is going to need further follow-up. So to summarize everything that I've done, the first question I looked at is, what is the contribution of known ALS genes in our cohort? We found mutations in 28% of our sporadic cases and 64% of our familial cases, although we're not really sure how many of these are actually pathogenic. There aren't really good criteria without having functional evidence of identifying which ones will cause disease. We didn't find any to be statistically significant. So there's going to be a lot of, we're going to need a lot of functional validation to determine which ones are pathogenic. We also found that mutations across multiple genes can act synergistically and decrease the age of onset. So that will also be interesting to see the functional effects of having multiple mutations in multiple genes at once in a cell culture model. But we also found that we're just going to need larger sample sizes to be able to detect statistical significance for rare variant associations. So for C9orf72, what characteristics are important for disease? The genetic background is very important, with one SNP promoting, well, the background marked by this SNP, we don't know that it's actually this SNP, it could be something else in this genetic background that's promoting expansion. And then this rare SNP that is mediating the effects of the expansion. And that could be either by, altering the expression, the methylation, the formation of RNA foci that are seen with the expansion. Also instability, both across families and across tissues. And the expansion size is not the only important thing to developing disease. We're going to need a lot more samples to look more closely at this. Southern blots are very hard to do, so that's maybe going to take a while. But also, is it possible that the effects of the expansion size are more to do with cell type, and we need to be looking more specifically at motor neurons versus just a chunk of spinal cord, which has a mix of different cell types. And then finally, can we identify novel genes that cause ALS? And we identified the rare mutation in TREM2 that was associated with sporadic ALS and was upregulated in ALS spinal cords. We're still not sure, what exactly is causing this effect. Is it because the mutation impedes its ability, the ability of the microglia to remove dead neurons, or is it that it's promoting a cytotoxic activation of the microglia and they're actually attacking healthy motor neurons? This is the neuroinflammatory pathways are a possible target for intervention in the future, and an additional source for candidate genes. So with that, a lot of people I'd like to thank. First, my thesis committee, especially Matt and Allison. As Matt told you, it was kind of a stressful situation with Bob Balo moving to California, and I'm very grateful that they both stepped up and let me stay and continue my work in Matt's lab. And Tim Miller for being a really great committee chair. Giving really great advice, and likewise with John and Christina and Joe. I'd like to thank the Harms Lab / former Baylor Lab. I kind of just threw you guys all together. Especially Jared. I know neither of us is particularly chatty, but it's been really great working with you. And all these other people. Special thank you to the Miller Lab for letting me perform radioactive experiments in your lab, and especially Amy and Kathleen, who ordered radioactive material and helped me dispose of it and everything. Done without you. And Erica Koval, who's not here anymore, but she did all the TREM2 expression studies in mice and patients. Also, the WashU neuromuscular group in general, and the Hope Center for their support financially and putting on really great seminars and discussion groups. The Goat and Kachaga labs, they did a lot with the genotyping studies, especially Carlos and Bruno, who were very involved in the TREM2 study. Renata Lewis, who taught me Southern blots, and gave very valuable advice on troubleshooting them, which is nearly impossible. And some other people, Gabe and Dave from the Gurnett lab, have been really great with helping us with our exomes, which I didn't actually talk about, but you were very helpful. Jeannie, our program coordinator, and the entire human and statistical genetics program. And then I'd like to thank my family, being awesome, and my brothers who come out every year at Christmas time to visit me. And I'm really glad that Jim and I didn't follow through on whatever we were plotting here. And then I'd like to thank my friends for making sure that PhD wasn't the only thing I got out of this experience. I also got 3 kickball championships. It's a lot of fun. Especially the Elizabeths, both Danka and Tuck. Particularly Elizabeth Danka for our awesome Halloween costumes. Best ever. So with that, I will take any questions. Gabe?
I was wondering if you have plans, maybe it's better question for that, or have tried to look over time in the same person and see if their expansions were so bad at the beginning or when they got bad.
Yeah, that's a really good idea. We don't.
You repeat the question.
Oh, he's wondering if we have looked at expansions across time in the same patient, if they, get larger as their disease progresses. We don't have those tissues. The only place we could look would be in blood, because we can't really take multiple brain samples. Other studies have looked at it, and they didn't really see any difference in size at different time points. I don't remember how far apart those time points were. Anyone else? So either I explained everything perfectly or I totally lost you. Yeah.
So in your Southern blots of families, there's the twins and there's all the expansion of both. Is there any difference in pathology, like, or between like pathology or large sciences between like the twin that had less than twin dog? Or
I don't think we've looked at any other differences between them. I just know one has ALS and one doesn't.
We don't have tissue from the one that
Yeah.
is still alive.
Right.
I'm sure your thesis committee members have quite additional questions. So again, 4 o'clock, the first floor conference room at Biotech for the party.